Hi, welcome to my presentation on the functional diversity of microbes. So last week we covered the metabolic diversity. We're going to be revisiting a good bit of that information now because metabolic diversity is part of functional diversity. So functional diversity means all of the different forms and functions that microbes can have, which includes, of course, their metabolism. That's a big part of their function. It also includes the roles they play in their environment or in their communities, and it includes their morphology, which is their shape and size, and the structures that they have. So a lot of the time, functional diversity kind of corresponds with phylogeny, or the evolutionary relationships between microbes, but it doesn't always align with the phylogeny. That's because sometimes you can have convergent evolution where the same trait evolves multiple times. So we're all familiar with an example of that in wings. Birds have wings, bats have wings, and insects also have wings. So that's three different types of wings that all evolved independently of each other. So birds, bats, and insects are not all closely related to each other just because they have wings. They all have different types of wings that evolved at different times, which is convergent evolution. So you can have that occurring with microbes as well, where a particular type of metabolism or a particular shape could evolve multiple times in microbes that are not exactly closely related to each other. You can also have traits that are lost through evolution if you have deletion mutations. Where a whole gene is deleted, then that trait that the gene coded for is just going to be lost. So you can see that with some algae, for instance, that don't do photosynthesis anymore, but they still have some of the machinery for photosynthesis. So they've lost some of the genes they need for photosynthesis, but they still have some of them, which means that we can still see that they used to have it and that they come from a lineage that had photosynthesis. But if all those genes were lost, then we wouldn't be able to tell. So sometimes that can happen, especially for traits that don't require as many genes as photosynthesis does. You can also have traits that are gained by horizontal gene transfer, which has played a large role in microbial evolution. So a lot of microbes will have, for instance, photosynthesis, even though they're not really closely related to each other, and it's because they picked those genes up through horizontal gene transfer from an unrelated microbe that already had those photosynthetic genes. So because of all those considerations, you're going to have some of these functional traits distributed in different phyla or different taxonomical classifications of microbes that are not really closely related to each other. And in the next chapters, we're going to move on to the phylogeny and talk a bit about that. So here's kind of a graphical summary of some of the most important prokaryotic phyla and important metabolic functions that they have. So a phylum is the level of taxonomic organization that is just under the domain. So a phylum is like the biggest grouping of bacteria you can have that's not just the domain bacteria, all bacteria, or the domain archaea, all archaea. Right under domain, you have the phyla. So in this diagram, is showing you prokaryotes. So in blue you have bacterial phyla, and then in red you have the archaeal phyla. And then these dots are all corresponding to different metabolic functions according to this key here. So you can see that dots of the same color, that's always the same function, and those dots are kind of widely distributed across phyla. So you can have bacteria that are not closely related to each other, that have the same metabolic function or that have the same function as archaea, even though they're not closely related. So some of those functions, well, basically all of these functions actually we covered in the previous chapter on metabolic diversity, and a lot of them we're going to talk about more today. So you have hydrogen oxidation. That one we're actually not really going to talk about today, but that is the use of hydrogen as an electron donor for respiration. So you can see that's very widespread. A lot of archaea and a lot of bacteria have that capability, or species in a lot of bacterial and archaeal phyla have that capability. Then you have the acetogens, which are bacteria that generate acetate. So you can see there's not that many of them, but they're around. Then you have methylotrophy, which is the using organic compounds that do not have carbon-carbon bonds for energy as electron donors. So that means basically organic compounds that only have one carbon in them. The most common examples would be methanol and methane. And then, as you may remember, there's a special type of methylotroph, which is the methanotroph. And the methanotroph uses methane as an electron donor. Methylotrophs can use methane and be methanotrophs, or they can use something else like methanol and be methylotrophs, but not also methanotrophs. But those are all considered together here in this diagram with the red dot, so you can see that's pretty widespread as well. Then you have nitrogen fixation, taking atmospheric nitrogen and converting it into ammonia. That is quite common. Actually, a lot of different archaea and bacteria are able to do that. Then you have denitrification, taking nitrate and reducing it as an electron acceptor to various nitrogenous gases like nitric oxide, nitrous oxide, and molecular nitrogen, which is in the pale blue dot, so that's pretty widespread as well. The nitrification would be taking ammonia and converting it to nitrate, using it as an electron donor. So you can see that's quite widespread as well. A lot of those phyla are clustered kind of together, which means they're more closely related to each other. Actually, though, down at this end of the diagram, the epsilon proteobacteria down through the beta proteobacteria, these are not phyla, actually. These are classes within the giant phylum Proteobacteria. So that's all together in one phyla. But you can see a lot of these guys have denitrification or nitrification. So those guys are all, fairly closely related to each other. But then it's also popping up at other ends of the diagram in prokaryotes that are not closely related. Then you have dissimilative iron oxidation and dissimilative iron reduction, using iron as an electron donor for oxidation or as an electron acceptor for reduction for respiration. Bacteria or archaea that do that would like to live, bacteria that reduce iron would like to live with bacteria that oxidize iron because they can kind of trade it off between each other. So those traits are also fairly widespread. You have a lot of archaea that are able to do one or the other of those. Then you have dissimilative sulfur oxidation, which goes along hand with dissimilative sulfate reduction and dissimilative sulfur reduction. In dissimilative sulfur oxidation, you are using hydrogen sulfide as an electron donor for respiration. In the different types of sulfur reduction, you're using either sulfate or sulfur as electron acceptor and reducing it to hydrogen sulfide. So those guys all like to live with each other as well. And you can see that is pretty widespread. All those traits are pretty widespread. Then finally, phototrophy. Oxygenic phototrophy is not widespread at all. It only exists here in the Cyanobacteria. Then you also have anoxygenic phototrophy, which is more widespread and has actually spread through different phyla by horizontal gene transfer. So most of these functions we're going to go into more detail with in the upcoming slides. So first we're going to focus on phototrophy in bacteria. So first we'll talk about the cyanobacteria, which, as we had mentioned before, are responsible for the Great Oxidation Event, or the oxidation of the Earth's atmosphere in response to rising oxygen levels, since all that oxygen was produced by the cyanobacteria through oxygenic photosynthesis. So that makes the cyanobacteria some of the most important bacteria on Earth, certainly the most important in the history of life on Earth, and still very important today. So all cyanobacteria are able to do oxygenic photosynthesis, but apart from that, there's a lot of diversity in that phylum. So they come in all shapes and sizes. These pictures are showing kind of some of those shapes and sizes. So they can form long filaments, they can form big ball-like clusters of cells. Sometimes in the filaments you can't even really see like where one cell ends and the next begins. Also whatever this is. And also just kind of free-living cells. A few cyanobacteria actually can switch to anoxygenic photosynthesis if they're not able to do oxygenic photosynthesis for whatever reason, like if there's not enough water around maybe. All cyanobacteria are autotrophs that use the Calvin cycle to fix carbon, but a few of them also can use organic compounds for their carbon source, which means that they would be behaving as heterotrophs when they're doing that. So they're all autotrophs, but a few of them can also act as heterotrophs and use some organic compound like glucose for a carbon source. A lot of cyanobacteria can also fix nitrogen in addition to fixing carbon. In general, they're going to be doing photosynthesis in the day, since that's when you have light. So they would be doing the light reactions of photosynthesis in the day. That's true for a lot of phototrophs, actually, since you only have light in the day. You can only do the light reactions in the day. Then at nighttime is when a lot of phototrophs will do their dark reactions. For cyanobacteria in the nighttime, they're going to get energy from fermentation or from aerobic respiration. So during the day, they would be doing both the light and the dark reactions, fixing their carbon dioxide into glucose and other carbohydrates. Then at nighttime, they can use that glucose in fermentation or in aerobic respiration as a source of energy. Usually cyanobacteria are blue-green because of their pigments. Their pigments are going to be chlorophyll and phycobilins. Other bacteria are going to use bacteria chlorophylls for their pigment. Only cyanobacteria and eukaryotes use chlorophyll. Actually, in the past, cyanobacteria were called blue-green algae because I guess scientists didn't know that they were a bacteria at that point, but they were just named for their color. When they're in long filaments like that, they can form really large masses that you can actually see, with your eyeball, which is why scientists figured that they were algae. That and the fact that they do photosynthesis. But they're actually bacteria. Some people still call them blue-green algae, though. A lot of the cyanobacteria use gliding motility to move along the surface, and they do phototaxis very frequently as well, meaning that they move towards light. And specifically, they'll move towards the wavelengths of light that chlorophyll is most able to absorb. And a lot of the time, they'll have gas vesicles, little membrane sacs full of gas inside the cell that they're using to control how deep they are in the water. That way, they make sure that they're not so deep that there's not enough light for them to do photosynthesis. So a little bit more about how cyanobacteria fix nitrogen for the ones that do. They're going to be using the enzyme nitrogenase, which we mentioned before. Actually, all nitrogen fixers are going to be using that nitrogenase enzyme complex. And as we mentioned, that enzyme is inhibited by oxygen. So cyanobacteria, of course, produce oxygen through oxygenic photosynthesis. The ones that fix nitrogen are going to need some type of a way to separate their oxygenic photosynthesis from their nitrogen fixation. Actually, nitrogenase is irreversibly inhibited by oxygen, which means that once oxygen binds to nitrogenase, it's not going to work and it will never work again. You're going to have to get rid of it and build a new one from scratch, which gets expensive. So different cyanobacteria will have different strategies for separating their nitrogen fixation from photosynthesis. Some of them will only fix nitrogen at nighttime. Some of them will kind of form a special cell type called this heterocyst. A heterocyst is larger than the cells around it, and it has really thick cell walls. The cell walls are thick enough that oxygen actually cannot get through them. So even though all the cells around this heterocyst are producing oxygen, the heterocyst itself does not have oxygen inside of it. So it's not going to be able to do the oxygenic photosynthesis either. It needs to not have oxygen inside of it. It's also not able to fix carbon dioxide because of that, so it's kind of relying on anoxygenic photosynthesis and relying on its neighbors for fixed carbon. So it's basically going to be trading the nitrogen that it has fixed with its neighbors in exchange for carbon that they have fixed. So here you have a picture of some filamentous cyanobacteria, and you have some heterocysts there. So you can see they're larger than the cells around them, and you can see the really thick cell wall that they have. They don't have all of those membrane invaginations, since they're not doing as much photosynthesis as their neighbors. And then in this diagram you can see that the neighboring cyanobacterial cells are fixing carbon and transporting those fixed carbon compounds like glucose into the heterocyst. The heterocyst, meanwhile, is fixing nitrogen and transporting that out as ammonia or as amino acids to its neighbors. So cyanobacteria like to live in water, especially warm water. They're very common in the ocean, also in other bodies of water. And they're very important in the ocean because of their ability to fix carbon and nitrogen. Actually, most life forms that live in the ocean are going to be getting their fixed carbon and nitrogen from cyanobacteria ultimately. Not necessarily directly from cyanobacteria. Some of those things will eat cyanobacteria and get the carbon and nitrogen. But some things are going to eat the things that eat the cyanobacteria. So all of this fixed carbon and nitrogen that the cyanobacteria are producing is going to filter up through the food chain to all organisms in the ocean. And of course, they're especially important in warmer waters and less so in the colder waters. They like to live as symbiotes with different types of creatures. Some of them will live in the ocean in symbiosis with diatoms. A diatom is a type of algae that has like a cell wall made out of silica that kind of looks like glass. In this picture here, you actually have several different examples of diatoms that have cyanobacteria in association with them. So these guys have cyanobacteria inside the cell. These guys have cyanobacteria stuck onto the outside of the cell. And you can see kind of the sharp edges of the diatom's silica cell wall there. You can also have cyanobacteria on land that live in lichens, which a lichen is kind of a composite organism that's always going to have a fungus in it and then an algae or a cyanobacteria. They can also live in symbiotic relationships with some plants. Not only are they important in the oceans, but they're also important in hot springs and in deserts. They're frequently the most common phototroph to have in a hot spring or in a desert, so they can also, they can, not only do they like it warm, but they can deal with hot temperatures. A lot of them can deal with hot temperatures. When it gets really warm, they can form blooms. A bloom is like a mass of some type of bacteria or algae in the water that is reproducing like so much that it's filling up this area of the water, and you can just see it with your eyes. So actually some of these blooms formed by the cyanobacteria can be visible from space. Here's an example of a bloom in one of the Great Lakes. All the green swirls here are cyanobacteria that are just reproduced to incredible levels here due to the warmer weather. So As we have climate change increasing the warmth of the planet and warming up our oceans, these blooms are becoming more common, which is an issue because some of them can be toxic. So some of these cyanobacteria can have toxic products that they make, and when you have that many of them in a bloom, it can become, dangerous for people who might be drinking that water, also dangerous for fish that would like to live there. So it can be very disruptive to the local ecosystem when they get that out of control. Also, you will sometimes have cyanobacteria in your water sources, and they kind of give the water an earthy flavor. So if you notice that type of flavor in the water, it might be due to cyanobacteria. Now that's not dangerous. The actual compound that gives it that earthy flavor is not dangerous at all. Next, we have the purple sulfur bacteria, which is our first group of anoxygenic phototrophs. All of the purple sulfur bacteria are in phylum Proteobacteria. There's two families of the purple sulfur bacteria in that phylum, which are the Chromatiaceae and Ectothyroidospiraceae. So I have a little summary here of the taxonomic hierarchy, starting with the phylum. So higher than that, you would just have the domain bacteria. So the phylum for these guys is Proteobacteria. Then you go into classes. Their class is Gamma Proteobacteria. Then orders. Their order is Chromatialis. Then families, which are the Chromatiaceae and the Ectothyroidospiraceae. And then you have the genera, which genera is the plural of genus, and the species. So family is kind of a lower taxonomic grouping. It's just above the genus. The purple sulfur bacteria are all going to be using hydrogen sulfide, or H2S, as an electron donor for their anoxygenic photosynthesis. Once H2S has donated its electron, it becomes just sulfur. Then they're going to store that sulfur in little granules, and then they can use it later on as an electron donor as well. So when they run out of hydrogen sulfide, they would have converted all of it to sulfur. Then they can turn around and use the sulfur as an electron donor and oxidize that to sulfate. They like to live in the water. They like to live in kind of places where you have the hydrogen sulfide. So that's kind of limiting what types of water they could live in. They also don't like to have a lot of oxygen in their environment, so that's further limiting where they can live. So you're going to find them living in lakes, but kind of towards the bottom of the lake, where you don't have oxygen penetrating. Also in sediments, which would be kind of the dirt basically at the bottom of some body of water. We call that sediment. So you're not going to have oxygen down there. Also in sulfur springs, where of course you have hydrogen sulfide in large amounts. So they need to live somewhere that has light. They can't be so deep that there's no light, but they also need to not be so shallow that there's oxygen. So if they're living in the sediment, they have to be kind of not that deep in the sediment so that light penetrates, but still there's no oxygen. A lot of the time they like to live with sulfur-reducing bacteria or sulfate-reducing bacteria, because those are the guys that produce hydrogen sulfide. They would either be reducing sulfur to hydrogen sulfide or sulfate to hydrogen sulfide. Then the purple sulfur bacteria will turn around and use that hydrogen sulfide as an electron donor, producing first sulfur and then again sulfate. Your purple sulfur bacteria are all autotrophs that use the Calvin cycle to fix their carbon. In accordance with their living habitats, a lot of them are going to be halophilic or alkylophilic due to those sulfur springs they like to live in. And they're purple in color because of their carotenoid pigments. So they also have the bacteria chlorophyll pigments, but their color is coming from the carotenoid pigments. So here you can see some examples. Well, all of this is actually examples of the purple sulfur bacteria. But in these pictures you can see the color that they make. Next we have the purple non-sulfur bacteria, which are also in phylum Proteobacteria. They are in the classes alpha Proteobacteria and beta Proteobacteria, and it's just a variety of genuses or genera in those classes. So these guys are not all closely related to each other. They're all in the same phylum, but apart from that, they're pretty distantly related, which means that they have shared the genes for photosynthesis amongst each other by horizontal gene transfer. So since, well, I guess, which makes sense since they're not closely related, they have a lot of diversity. So you can see in the picture here all the different, well, some of the different shapes they can form. So they have a pretty wide range of variation there as far as their shape. And they also have a lot of metabolic diversity. Of course, they all do anoxygenic photosynthesis, but apart from that, they have a lot of diversity, metabolically speaking. Usually they're photoheterotrophs. Some of them are photoautotrophs, but most of them are photoheterotrophs. They use all types of organic compounds as carbon sources and as their electron donors. So the photoheterotrophs would be using the Calvin cycle, and for an electron donor they would be using something inorganic, either molecular hydrogen or hydrogen sulfide or iron. They're doing anoxygenic photosynthesis for energy during the day. In the nighttime, they're going to switch to something else. Some of them are going to do aerobic respiration. Some of them will do aerobic respiration using an organic compound as the electron donor. Some will use an inorganic compound as electron donor, which would mean that they're living as chemolithotrophs in the dark. Some of them will switch to fermentation. Some of them will switch to anaerobic respiration. Most of them do fix nitrogen, but not all of them. They like to live in the water. You'll find them in lakes, usually close to the bottom in the sediments. They also live in sewage, and usually they are purple, red, or orange due to their carotenoid pigments, although in the picture you can see that none of these are actually purple. The blue one here and then the very yellow one on the end, those are both mutants, so they're missing some of their carotenoid pigments. This one is actually missing all of the carotenoids, so you can just see the color of the bacterial chlorophyll in there. All of them would actually have that bacterial chlorophyll, but then they also have a variety of different carotenoid pigments that are giving them their orange, red, or yellowish colors. And some of them are actually purple. Then you have another group that's very diverse called the aerobic anoxygenic phototrophs. These are also in classes alpha proteobacteria and beta proteobacteria in the phylum Proteobacteria. They also don't share a recent common ancestor, so they're kind of scattered throughout those two classes in different genera throughout those classes. And they've also shared their genes for photosynthesis by horizontal gene transfer. They are also photoheterotrophs. Actually, most of them are just doing photosynthesis as sort of a side game. It's a place where they can get energy when they're not able to get enough energy from their aerobic respiration. Then they'll turn to photosynthesis to supplement. So they're not doing photosynthesis as their primary source of energy. None of them are autotrophs. As their primary source of energy, they would be doing, as the name implies, aerobic respiration. So these guys have to live in the presence of oxygen, and they'll use organic compounds as their electron donor for aerobic respiration. They like to live in the ocean, usually along the coasts. Their colors are going to range from yellow, orange to pink, also due to the carotenoid pigments that they have. Some of the main differences that kind of define them as separate from the purple non-sulfur bacteria is that they are never autotrophs, whereas the purple non-sulfurs can be autotrophs sometimes. And most importantly, they only live in environments that have oxygen. The purple non-sulfur bacteria, some of them will live in environments that have oxygen if they're trying to do aerobic respiration at nighttime. Some of them will live in anoxic environments if they're trying to do fermentation or anaerobic respiration in the nighttime. Your aerobic anoxygenic phototrophs will only ever live in environments that have oxygen because they require oxygen for aerobic respiration, which is their primary source of energy. Photosynthesis is just a supplement. Then you have your green sulfur bacteria in phylum Chlorobi, which means that these guys actually are all closely related to each other. They are all going to use hydrogen sulfide as their electron donor for anoxygenic photosynthesis, which means that they're oxidizing that to sulfur. So similar to the purple sulfur bacteria, they're also going to use the hydrogen sulfide, oxidize it to sulfur, deposit those sulfur atoms as granules outside the cell this time. Purple sulfurs will usually deposit it inside the cell. And then once their hydrogen sulfide sources are running low, they're going to turn around and use that sulfur as an electron donor, which oxidizes it to sulfate. These are also autotrophs, but they're fixing carbon using the reverse citric acid cycle, not the Calvin cycle. And they have a special structure in them called the chlorosome. You can see some chlorosomes here. So they're usually just underneath the cell membrane. There's these kind of like membrane-bound cylinders formed by folds of the cell membrane, and they're just full of pigments, just packed full of pigments. So they're able to absorb a lot of photons in those chlorosomes, and then each chlorosome is going to funnel all of those photons in towards a photocenter where photosynthesis would actually be taking place. So because they're using these chlorosomes to gather light like that, they're able to live and do photosynthesis even in places where there's barely any light at all. Out of all the phototrophs that exist, the green sulfur bacteria require the least amount of light. So you'll find them living in lakes or in the ocean, like deep lakes. They're going to live very far down. So they like to have places that don't have oxygen and barely any light. They're kind of specialized to do better at light levels that no other phototroph can handle. So if you go to kind of a higher light level, then there's going to be all types of phototrophs there competing for the light, but these green sulfur bacteria are going to go down below that to a level of the water where there's barely any light at all. No other phototroph will be there because there's not enough light for them. It'll just be the green sulfur bacteria. You can actually find them at the very bottom of the ocean where you would think that there's not any light at all, but they would be living near some of those deep sea vents. They're living near some of those deep sea vents that produce a lot of heat, and that heats up the rocks around the vents, and then those rocks emit infrared radiation. So they're actually able to absorb those infrared photons and do photosynthesis just with that tiny bit of glow that's coming off of those hot rocks at the bottom of the ocean near those deep sea vents. Usually they're not motile, so they're just kind of floating around in the water, and they would be usually green, but they could also be brown. It's a combination of the bacterial chlorophyll and carotenoid pigments that are giving them those colors. So here you can see some examples of the green sulfur bacteria, and then here are the colors. Down here you have a brown example, and then here's a green example. It does look mostly yellow to me, but that yellow is a little bit green. You can find green sulfur bacteria in special types of symbiotic relationships with different types of chemoorganoheterotroph bacteria. They're forming an arrangement that's called a consortium, where you have the chemoorganoheterotroph in the middle, and then around that in kind of a shell, you have the green sulfur bacteria. So here is an example of that. This would be the central chemoorganoheterotroph, and then you have the green sulfur bacteria around it in a circle, and you can see their chlorosomes here. And then here you can see that again. So here you have a chemoorganoheterotroph in the middle, but it's not really stained, and you can see the green sulfur bacteria clustering around it. Here you have chemoorganoheterotrophs that are like coated by these green sulfur bacteria. And then here you have another stain that is only showing the green sulfur bacteria, but you can just see the outline of the chemoorganoheterotroph that's inside of them. So those are all examples of consortia. Then you have the green non-sulfur bacteria, which are in class Chloroflexi within the phylum Chloroflexi. So these guys are also closely related to each other. They're not very well studied though. We don't know that much about them. Usually they're photoheterotrophs. They'd be using different organic compounds as their electron donors. but they're also able to switch to living as photoautotrophs, in which case they would be using either molecular hydrogen or hydrogen sulfide as their electron donor for their carbon fixation reactions, which would be the 3-hydroxypropionate bicycle. When it's dark, they will switch to aerobic respiration, so they're going to live in the water again, in lakes and springs, but kind of higher in the water where you have oxygen still. Some of them are thermophiles, which would be, the ones that live in the springs. Most of them are going to use gliding motility, and usually they're going to be green, not always, but because of their bacterial chlorophyll pigment, they'll usually be green. So here's some examples of the colors. So usually green, sometimes not so green, and here's some examples of what they look like. So these would be the green non-sulfurs. These guys actually kind of look more like brown, and sometimes they can form these long filaments. Then you have the Heliobacteria, which live in, which are part of family Heliobacteriaceae. That's actually in the phylum Firmicutes. Within the phylum Firmicutes, you have class Clostridia, then order Clostridialis, then family Heliobacteriaceae, which would be the Heliobacteria. So the rest of that class and order are not phototrophs, just the family Heliobacteriaceae. So these guys are closely related to each other. They're also photoheterotrophs that would use organic compounds as their electron donor. In the dark, they are going to switch to fermentation. They're also nitrogen fixers, and they live in the soil. They really like wet soils, and the most common place to find them is in rice paddies. So you have an example of a rice paddy here. So you can see these paddies are usually pretty soaked with water, so the soil is very wet. And apparently in Japan they actually have found a way to make like art with their rice paddies. So this is Naruto. The Heliobacteria are really important for those rice paddies and for rice production because they fix nitrogen for the rice. So without the Heliobacteria in there fixing nitrogen, the rice aren't able to get enough nitrogen to grow well. Some Heliobacteria are alkalophiles and they like a high pH environment. They're also able to produce endospores, which is pretty unusual. So most bacteria cannot produce endospores. The ones that can, about half of the ones that can are going to be found in class Clostridia. There's also another type of bacteria that's important that has a very similar name but is very different, which is Helicobacteria. So Helicobacteria you probably haven't heard of specifically before, but you probably have heard of H. pylori, which is Helicobacteria pylori. So that's the bacterium that's responsible for stomach ulcers. That's very different from Heliobacteria, even though the name's very similar. Then we have just a couple of other examples of anoxygenic phototrophs that have been kind of recently discovered. So we found phototrophic Acidobacteria in the phylum Acidobacteria. We don't know of many. There's just really one species that we have discovered in that phylum that is a phototroph. It's a photoheterotroph that uses fatty acids as a carbon source. During the nighttime, it's going to switch to aerobic respiration, and it was found living in Yellowstone hot springs, so it's thermophilic. Then you also have another species in phylum Gemmatimonidates that was recently discovered that's a phototroph. It was discovered in a lake in the desert, in the Gobi Desert, which is a very large desert in Asia. It's also a photoheterotroph, and it's also similar to the aerobic anoxygenic phototrophs, it's also using photosynthesis as a secondary energy source. Its main source of energy is aerobic respiration, and then it's just kind of supplementing that with anoxygenic photosynthesis. So since it's using photosynthesis as a supplement to its main source of energy, we think that it got photosynthesis from horizontal gene transfer also. So that's phototrophy. Now we'll talk about the sulfur cycle, which basically means sulfur oxidizers and sulfur reducers. So first we have the dissimilative sulfate reducers. Then you also have dissimilative sulfur reducers on the next slide. These are called dissimilative reducers because they are reducing sulfate in order to get energy through their metabolism. They are not reducing it in order to incorporate sulfur into like the amino acids that need sulfur. So they're not reducing sulfur in order to build molecules for the cell. If they were reducing sulfate or sulfur in order to build molecules for the cell, that would be assimilative sulfate reduction or sulfur reduction, which actually a lot of different microbes can do. It's dissimilative sulfate reduction because they are using the sulfate reduction for energy, which is much less widespread. There's not that many bacteria that can do it. You actually do have 30 different genera that can do dissimilative sulfate reduction in 5 different phyla, one of them an archaeal phylum. So that is a lot, but compared to the overall amount of bacteria that are out there, it's not that much. So for archaea, the dissimilative sulfate reducers are in the genus Archaeoglobus in phylum Euryarchaeota. For bacteria, most of those genera are in class Delta Proteobacteria, which is in the phylum Proteobacteria. And then the diagram at the bottom is showing you the different genera of dissimilative sulfate reducers. So here you have the Archaeoglobus, which is your archaeal genus that can do sulfate reduction for energy, and then the rest of these are all bacterial. You have a genus in the phylum Thermodisulfobacteria, another genus in the phylum Firmicutes, another genus in phylum Nitrification, and then all of these other genera are all in phylum Proteobacteria, class Delta Proteobacteria. So it's a very diverse group, as you might guess from how many different phyla include sulfate reducers. What they all have in common is that they will use sulfate as their final electron acceptor in anaerobic respiration, which then reduces it to hydrogen sulfide or H2S. Actually, some of them can use other things. For instance, some of them can use nitrate as their final electron acceptor. Some of them could also use fermentation or switch to fermentation instead of anaerobic respiration, but they're all able to use sulfate as their final electron acceptor. Most of them are going to be obligate anaerobes, so they cannot tolerate the presence of oxygen. For their electron donor, they would usually be using molecular hydrogen or some type of organic compound. So if they're using molecular hydrogen as their electron donor, then they are chemolithotrophs. If they're using organic compounds as the electron donor, then they are chemoorganotrophs. And some of them are also actually going to be autotrophs, which would be fixing their carbon through some carbon fixation pathway. You have 2 different groups of the sulfate reducers. Some of them are complete oxidizers, which would be completely oxidizing their organic electron donor to carbon dioxide. Some of them are incomplete oxidizers, which would not completely oxidize their organic electron donor to carbon dioxide. And of course, the ones that are using molecular hydrogen as an electron donor wouldn't fit into either of those groups. So the dissimilative sulfate reducing bacteria or archaea will like to live in the water or in the soil. They can live in sediments at the bottom of the ocean. They're going to live in places where sulfate is common though. Some of them are actually thermophiles. They'll be living in springs or at the bottom of the ocean near deep sea vents. A lot of the time you'll find them living in community with the purple sulfur bacteria or the green sulfur bacteria. Both of those are using hydrogen sulfide as an electron donor, and the dissimilative sulfate reducers produce the hydrogen sulfide for them. They could also live with the dissimilative sulfur oxidizers, who will also use the hydrogen sulfide that they produce. Then you have the dissimilative sulfur reducers. Those are quite common. The main difference is that the sulfate reducers reduce sulfate to hydrogen sulfide. The sulfur reducers reduce sulfur to hydrogen sulfide. There's not quite as many genera of the dissimilative sulfur reducers. There's 25, and again in 5 different phyla. For Archaea, all of the genera of sulfur reducers are in phylum Crenarchaeota. For bacteria, again, most of them are in class Delta Proteobacteria in the phylum Proteobacteria. They're also very diverse, actually even more diverse than the sulfate reducers. They're all able to use sulfur as their electron acceptor for anaerobic respiration, which would reduce it to hydrogen sulfide. But a lot of them can also use other things like nitrate or even other things besides, or do fermentation. Most of them are, again, obligate anaerobes that can't tolerate the presence of oxygen. They're usually going to use molecular hydrogen or some type of organic compound as their electron donor. And again, if they're using molecular hydrogen, that makes them a chemolithotroph. If they're using an organic compound, that makes them a chemoorganotroph. Some of them are actually going to be facultative aerobes, though, which would be bacteria that can switch to using oxygen as their final electron acceptor if it's available. Again, we can divide them into complete oxidizers and incomplete oxidizers, at least for the ones that are using organic compounds as an electron donor. The complete oxidizers would completely oxidize that compound to carbon dioxide. The incomplete ones would not oxidize it all the way to carbon dioxide, and it would remain as some type of oxidized organic compound. In terms of where they live, it's very similar to the dissimilative sulfate reducers, so they like to live in lakes, the ocean floor, or soil, in places where you have a lot of sulfur. And they're also going to be found living with the green and purple sulfur bacteria and with the dissimilative sulfur oxidizers, since all those groups are able to use the hydrogen sulfide that the sulfur reducers produce. Finally, you have the dissimilative sulfur oxidizers. Those guys are oxidizing sulfur for their metabolism to get energy. That makes it dissimilative. Assimilative sulfur oxidizers would be oxidizing sulfur in order to incorporate sulfur into their organic molecules in order to build the structures for the cell. So that's quite common. Dissimilative sulfur oxidation, oxidizing sulfur for energy, is much less common. Bacteria that can do this are usually found in 4 different, prokaryotes, I should say, that can do this are usually found in 4 different phyla. For Archaea, all of them are going to be found in phylum Crenarchaeota. For bacteria, again, most of them in phylum Proteobacteria. This is also a very diverse group, as you might expect from how many phyla examples of this type of metabolism are found in. They're usually going to be chemolithotrophs that are using sulfur or hydrogen sulfide as their electron donor for respiration. Actually, usually they're doing aerobic respiration. They could also use other sulfur compounds like thiosulfates, and some of them can switch to chemoorganotrophy as well, which would mean that they can switch to using organic compounds as their electron donor. Usually they're obligate aerobes, which actually makes a problem for them because hydrogen sulfide, their preferred electron donor, will actually spontaneously oxidize in the presence of oxygen, which would mean that they're not able to harvest the electrons from that. So if you have hydrogen sulfide just spontaneously oxidizing, that means the electrons will just directly transfer to oxygen, whereas these bacteria need those electrons to enter the electron transport chain so that they can use the energy of the electrons to pump protons and get energy. So if the electrons are not going through the electron transport chain, then the oxidation of hydrogen sulfide is useless to them. So they need some way to separate the oxygen that they're using as their final electron acceptor from the hydrogen sulfide that they're using as their electron donor. So they have different ways of doing that, but some of them would do it by living in symbiotic relationships with critters at the bottom of the ocean, like worms and crabs. They like to live in sediments at the bottom of the ocean. They'll also live in springs, basically places where you have sulfur. those sulfur compounds that they need, the sulfur itself or the hydrogen sulfide or the other sulfur compounds that they can use, some of them are going to be provided by geothermal activity depending on the location. So when they're living in a spring or near a deep sea vent, it's geothermal activity that's ultimately providing the sulfur for them. But it could also be that the sulfur is in a different format and it's being converted to sulfur, to actual sulfur, or to hydrogen sulfide by dissimilative sulfate reducers or sulfur reducers. A lot of them are autotrophs that use the Calvin cycle to fix carbon, and some of them are acidophiles. The ones that live in springs would be acidophiles. So here you have a couple examples of their living environments. This would be a spring, and this is a deep sea vent. And you can see all these kind of colorful stretches of the rock. Those are actually mats of these sulfur oxidizing bacteria. Now we'll talk about the nitrogen cycle, which includes nitrification, nitrogen fixation, and denitrification. So first we'll talk about nitrogen fixation. There's a special word for microbes that fix nitrogen, which is a diazotroph. So diazotrophs are microbes that fix nitrogen by converting molecular nitrogen from the atmosphere to ammonia or NH3. This process always requires the enzyme complex nitrogenase, which is irreversibly inhibited by oxygen. So every diazotroph has some way of separating oxygen from nitrogen fixation. A lot of them are going to be anaerobes, so they don't encounter oxygen anyways. The ones that are aerobic will have some mechanism for separating oxygen from their nitrogen fixation activities. Nitrogen fixation is an assimilative process, which means that these microbes are fixing nitrogen, converting it to ammonia in order to add that ammonia as amino groups onto amino acids or onto nucleotides so that they can build the organic molecules they need for the cell. In general, it's just that they're going to fix too much. They're going to make more ammonia than what they actually need for their macromolecules that they need to build, and then the extra is going to end up being exported outside of the cell, and other organisms that are around there can pick that ammonia up and use it to build their own macromolecules. So nitrogen fixation is a pretty widespread activity. There's 10 different phyla of prokaryotes that can do nitrogen fixation. That includes one archaeal phylum, which is Euryarchota. So not every species in that phylum or in this phyla are able to do nitrogen fixation, of course, but just a lot of them are. And you also have some phototrophs that can do nitrogen fixation, like we had mentioned. The Cyanobacteria, a lot of them can do nitrogen fixation. Also the purple non-sulfur bacteria and the Heliobacteria. A lot of these nitrogen fixers are symbiotes that live in these symbiotic relationships with plants or with fungi or with algae or even with worms, so simple animals. In that type of arrangement, the host is always benefiting by getting a regular source of fixed nitrogen, whereas the diazotroph is getting a nice environment, basically a nice place to live. And usually the host will be helping them keep oxygen out. So for instance, you have those root nodules, which are shown in the picture right here. Right, so this is the root of a legume, which legumes are like bean plants. And then these are the root nodules. So these are clusters of diazotrophic bacteria that are growing on those roots. They're actually encased in a little structure that's produced by the root, which is preventing oxygen from entering them. So they're being protected from oxygen there. That way they can keep fixing nitrogen without nitrogenase being inhibited. And it's also providing, just stability in terms of pH, water content, glucose availability. It's just a nice stable environment to live. A lot of other diazotrophs are going to be living in anoxic habitats, so places that don't have oxygen. A good example of that would be a sediment at the bottom of a lake or the ocean. Usually they're going to be obligate anaerobes, so they can't tolerate the presence of oxygen anyways. And some have other solutions, like the Cyanobacteria that use their heterocysts sometimes to keep oxygen out. We also have the genus Azotobacter, which also lives in oxic environments and is going to be using thick slime layers to keep oxygen out. So that's shown in these pictures here. These little dark streaks are actually the cells of Azotobacter, and you can see these kind of like gray capsules around them. That's actually the slime layer they produce, so oxygen's not able to penetrate through that. Then here you have colonies of that Azotobacter growing. You can see kind of how slimy they look. Next we have the nitrifiers, which are bacteria or archaea that are oxidizing ammonia or nitrite to nitrate. So they'd be using either ammonia or nitrite as their electron donor in respiration, usually aerobic respiration, and therefore oxidizing it. If they're using ammonia, that gets oxidized to nitrite. If they're using nitrite, that gets oxidized to nitrate. So usually these are obligate aerobes, and they're usually also autotrophs that use the Calvin cycle to fix nitrogen. A lot of the time they're going to have kind of elaborate folded internal membranes or membrane invaginations. So you can see that in these pictures here. So these are all membrane folds that are folded from the cell membrane, and here as well. So those membrane folds have the enzymes that they're using for nitrification. So their electron transport chain and also the other enzymes that they require to do that process and oxidize either ammonia or nitrite. There's two different types of nitrifiers. They can either be ammonia oxidizers or they could be nitrite oxidizers, like we mentioned in the previous chapter. If they are ammonia oxidizers, then they are using ammonia as their electron donor and oxidizing it to nitrite, which is NO2-. If they are nitrite oxidizers, they are using nitrite as their electron donor and oxidizing it to nitrate, which is NO3-. So for the complete process of nitrification, which is actually oxidizing ammonia to nitrate, you would need both the ammonia oxidizers and the nitrite oxidizers. They can only do one or the other step. Ammonia oxidizers are more diverse. So you have 2 bacterial phyla and 1 archaeal phylum that has the ammonia oxidizers in it. For the bacteria, ammonia oxidizers are in phyla Proteobacteria and Nitrispirae. For archaea, they're in phylum Thaumarchaeota. In general, the genera where you actually have the ammonia oxidizers are going to have their name starting with nitroso. So if you see the name of a genus and it starts with nitroso, that's probably a genus of ammonia oxidizers. For the nitrite oxidizers, on the other hand, you actually don't have any archaea that can do that process. They're all bacteria. They're again found in phyla Proteobacteria and Nitrispirae, and the genus names will start with nitro usually. So if you see a bacterial genus whose name starts with nitro, that probably means that those are nitrite oxidizers. There's just one exception for genus Nitrispira in phylum Nitrispirae. Some species in genus Nitrispira can do both ammonia oxidation and nitrite oxidation all in one bacteria. Apart from that, all the genera whose names start with nitro are nitrite oxidizers. For the ammonia oxidizers, the bacterial ones like to have a higher ammonia content in their environment. So you're going to find them living in the soil and the water wherever you have a lot of ammonia, which would be basically wherever organic matter is decaying. As living things are, dying and their cellular components break down, all those amino acids and nucleotides that have amino groups in them are going to release those amino groups as ammonia into the environment. So decaying organic matter is a great source of ammonia. Of course, decaying organic matter is very common in sewage, so you're going to find a lot of bacterial ammonia oxidizers in sewage. Archaeal ammonia oxidizers don't like to have a whole lot of ammonia in their environment. They like a more moderate or more limited amount. So you'll find them in the soil as well, just not as likely right by where something is decaying. And you'll also find them living in the ocean. And they especially like to live in acidic soil, so they're more likely to be acidophilic. The nitrite oxidizers would also be found living in the soil a lot, or in the water. They like to live with the ammonia oxidizers, which are a source of nitrite, which they need for their metabolism for an electron donor. And again, you have some of them that like to have a lot of nitrite in their environment and others that like to have less. Then finally, you have the denitrifiers. The denitrifiers are using nitrate or nitrite as an electron acceptor for respiration, which is usually anaerobic respiration. Then once that nitrite or nitrate has been reduced, then it's going to be reduced all the way to a gas, either nitrous oxide, nitric oxide, or molecular nitrogen. So there's 8 different phyla of prokaryotes that can do this process. 2 of them are archaeal phyla, the other 6 are bacterial. And it's so widespread in different phyla that probably those genes have been traveling through different prokaryotic species by horizontal gene transfer. So there's a lot of diversity among the denitrifiers as well. A lot of them are chemoorganoheterotrophs though, and a lot of them are facultative aerobes. So even though when they're using nitrate or nitrite as an electron acceptor, that does mean that they are doing anaerobic respiration since it's not oxygen. But a lot of them would be able to switch to using oxygen if it became available. They're very important for agriculture. They're actually not a good thing for agriculture because farmers are usually trying to add ammonia to the soil as part of their fertilizer, and the denitrifiers are taking it away. So first you would have your nitrifiers converting that ammonia to nitrite and then to nitrate, and then these denitrifiers, if they're there, will take that nitrite or nitrate and convert it into some type of nitrogenous gas, and at that point it leaves the soil. So as long as it's staying in the form of ammonia or nitrate or nitrite, just something like that, it's going to stay in the soil, so that's okay. But if it's being converted to a gas and leaving, now you need to add more fertilizer to replace what was lost. It's also a problem because the N2O they make is a greenhouse gas, and also the NO they make is partly responsible for acid rain. So overall, if you have too much of them, they're going to have a negative impact on the environment. So now we'll go through some other functional groups of bacteria, including the iron reducers and iron oxidizers and the methylotrophs, and then moving past metabolism at last. So first, the dissimilative iron reducers, who again are going to be reducing iron in order to get energy from it, not in order to incorporate it into their organic molecules that require iron to function, which would be assimilative iron reduction. So the dissimilative iron reducers, of course, are going to be using iron as a final electron acceptor to reduce it, but they also don't necessarily need to use iron. They could use other things as well. Usually they could also use manganese, so they would either be using Fe3+, or manganese 6+ as an electron acceptor and reducing it. They could also use other metals or other metalloids sometimes, but it's less common. And a lot of the time, they're going to be using either molecular hydrogen or some type of organic compound as their electron donor, which would be similar to the sulfur and sulfate reducers. If they're using molecular hydrogen, that makes them a chemolithotroph. If they're using an organic compound, that makes them a chemoorganotroph. And since they're always using some type of metal or metalloid as their final electron acceptor, that means they're doing anaerobic respiration. So they're pretty diverse, I guess, or flexible in terms of what they can use. Most of the dissimilative iron reducers can use a range of different compounds as their electron acceptor and their electron donor. Just depends on what's available in their environment. They're pretty diverse. There's 6 bacterial phyla that include iron reducers, and 1 archaeal phylum, which is the Crenarchaeota. And it's probably an ancient process. So we think that it evolved very early in the evolution of life. In general, the final electron acceptor for iron reducers is going to be some type of solid thing. So if they're using iron or manganese, that's a solid substance. And so they need to, figure that out somehow. So a lot of the time they're going to kind of harvest electrons off of this solid substance that's outside of their cells, and then those electrons will enter the electron transport chain, but the iron itself or the manganese itself will not enter the cell. It remains outside the cell, and they just kind of harvest electrons off the surface of it. Sometimes they would actually use phyla to attach to that metal and harvest the electrons. Most of them are obligate anaerobes, so they can't tolerate the presence of oxygen, and most of them are heterotrophs, although there are a few exceptions. You do have a noticeable number of them that are facultative aerobes that could switch to using oxygen as their final electron acceptor if it's available. But they usually live in places where you don't really have oxygen, like in sediments and deep underground, so in the soil, but like real deep. A lot of them are actually thermophiles, and they would be living wherever you have geothermal activity deep underground. So in these images, on the first one, you can see the results of an assay for iron-reducing bacteria. So you would add the iron reducers to a tube containing this substrate for them to grow on and use it as an electron acceptor, which would be iron, but also it's kind of, it's bound up in other compounds. So it's an iron compound, not pure iron. Then through their metabolism, they will be producing iron 2+. So when you reduce Fe3+ by adding an electron to it, you get Fe2+, which is magnetic. And so that's going to be attracted to this magnet. So when you see that the stuff in the tube is becoming magnetic, then you know that you have iron reducers living in there. And here you have a couple examples of iron reducers. This one is using its pilus to transfer not like individual electrons, but little bits from the substrate that it's growing on, and then it's going to strip the electrons off of those once it gets to the cell membrane. To go with your dissimilative iron reducers, you have dissimilative iron oxidizers who are using Fe2+ as an electron donor for respiration. So the dissimilative iron reducers are using Fe3+ and producing Fe2+. The iron oxidizers use iron Fe2+ and produce Fe3+ by stripping an electron from it, since it's the electron donor. There's a lot of diversity amongst the iron oxidizers. That includes 5 different bacterial phyla and 2 archaeal phyla. They usually live in places that either have low oxygen or low pH, or both, because if you have Fe2+ iron in the presence of oxygen, it will spontaneously react to form rust or iron oxide compounds if the pH is neutral or high. So in order to avoid that happening, you either need to have no oxygen or you need to have a low pH if there's oxygen there. So some of these guys are aerobic acidophiles, so they're going to be using oxygen as their final electron acceptor, but since they're living at a low pH as acidophiles, they avoid the iron that they're using as their electron donor being spontaneously oxidized by oxygen. You would find those guys living near certain springs or in the drainage zones for certain mines. Then you also have microaerophilic neutrophiles, so iron oxidizers that live at a neutral pH, but they also live in environments that have very little oxygen present. So you would usually find those living in like wetlands. So since they're microaerophiles, they are doing aerobic respiration using oxygen as their final electron acceptor, but they're only exposed to a very small amount of oxygen. So you will have some of the iron that they're using as their electron donor spontaneously rusting away, but most of it will not. And then you have the anaerobes. Some of them will like a neutral pH, some of them will like an acidophilic pH. It just depends. They're all going to be living in places where you don't have oxygen present. So usually in a wetland or in a sediment at the bottom of a lake or an ocean. You actually have some denitrifiers that are also dissimilative iron oxidizers, and you have some phototrophs that are dissimilative iron oxidizers. That would be specifically some of the purple and green sulfur bacteria. So those guys would be using iron as their electron donor when they do photosynthesis. Some of the iron oxidizers are autotrophs and some of them are heterotrophs. The last metabolic grouping that we'll talk about is the methylotrophs and methanotrophs. Methylotrophs being, again, microbes that use organic compounds that do not have carbon-carbon bonds as electron donors for getting energy. Methanotrophs are a type of methylotrophs that are using specifically methane as their electron donor. So methylotrophs are going to use always an organic compound, but usually an organic compound that only has one carbon in it, since that's generally the only way it would be an organic compound that doesn't have carbon-carbon bonds. So that usually means methane or methanol. So if you're using methane as your electron donor, you are a methanotroph and a methylotroph. If you're using methanol, then you are only a methylotroph, not a methanotroph. In general, methylotrophs and methanotrophs would be using that compound, either methanol or methane, not only as their electron donor for respiration, but also could be using it for their carbon source. There's 5 different bacterial phyla that include methylotrophs and 1 archaeal phylum. Methylotrophs can be aerobic or anaerobic. If they are aerobic, then they're going to be living in the soil and water wherever you have oxygen. If they are anaerobic, they would be living in sediments at the bottom of lakes or oceans where you do not have oxygen. They're usually pretty diverse in terms of what they can use for their metabolism. So they were going to use methane or methanol usually, but they could also use other things, other organic compounds, even inorganic one carbon compounds sometimes. And a few of them are also denitrifiers that would be using nitrite as their final electron acceptor. Focusing in on the methanotrophs, a lot of methanotrophs are going to have those membrane invaginations that have enzymes that they need for methanotrophy. Specifically, those enzymes are needed to oxygenate methane, which produces methanol, which is the first step in using methane as an energy source. So here you can see a couple different arrangements of those folded internal membranes that methanotrophs have. A lot of them will also have carotenoid pigments that make them look pink. So you can see that here. So these are colonies of those methanotrophs. You can see they have different pinkish colors. This is actually the imprint left by a leaf. So you basically get the underside of a leaf and press it down onto this large agar plate and see what grows, and it's, these pink methanotrophs. So a lot of the time you'll find them living on leaves as well. They're also very unusual because their membranes have sterols in them, which is not like bacteria, but it is like eukaryotes. So every eukaryote has some type of sterol in their membrane. If you're an animal, then you have cholesterol in your membrane. If you're not an animal, you would have a different type of sterol. And if you're a bacteria, if you're a bacteria, you don't have sterols in your membrane unless you're a methanotroph. Ecologically, they're very important because they use methane, converting it to other compounds. So methanogens would be bacteria that produce methane, Methanotrophs are bacteria that use methane, or archaea that use methane. Methane is a very powerful greenhouse gas, so it really traps heat very well, actually much better than carbon dioxide does, so it's much worse for our environment than carbon dioxide is. In terms of its potency. On the other hand, methane doesn't last very long in the upper atmosphere, whereas carbon dioxide lasts for hundreds of years. So overall, carbon dioxide is definitely the bigger threat there, but methane is also a significant threat. So these methanotrophs can help us address that by taking that methane out of circulation, converting it to something else before it reaches the upper atmosphere. You also have methanotrophs that live in symbiotic relationships, usually with different types of shellfish on the ocean floor. So down there on the ocean floor, you have certain places where methane is leaking up into the ocean from basically underground methane cisterns. And you're going to have methanotrophs living in those environments with usually symbiotically with shellfish or other things. So now we'll talk about some other types of functional groupings, not metabolic groupings. First, predators. There's not that many bacterial predators, but there are bacterial predators, which would be just bacteria that eat other bacteria or archaea, or even maybe protists, but usually not because they'd be too big. Most protists would be too big, that is. So bacteria are usually going to be chemoorganoheterotrophs, which means that they are getting their energy from organic compounds that have come from some other creature, and they're also getting their carbon from that same source. But that doesn't mean that they're predators. They're usually just more like scavengers or decomposers. So they're just kind of degrading dead things, or picking up the organic compounds that they find lying around in the environment. Bacterial predators specifically go after other microbes and eat them for their organic molecules. There's 3 different phyla of bacteria that include predators. Of course, not everybody in those phyla would be a predator, only some. And you've got 4 different kind of types of bacterial predators. You have the epibiotic predators. Those are going to attach to the prey microbe and suck nutrients right out of it. Then you have cytoplasmic predators that will actually go inside of their prey and start dividing inside of the prey and end up eating it from the inside out. Then you have paraplasmic predators that would prey on gram-negative bacteria. They're pretty similar to the cytoplasmic predators, but instead of actually going inside the cell membrane, they would just stay in the periplasm, get through the cell wall and stay in the periplasm and eat, divide in that periplasm and eat the prey cell from there. Finally, you have social predators that will swarm their prey in a group and eat it all together. One of the most interesting types of predatory bacteria is the Myxobacteria, which are found in order Myxococcales. These guys will swarm, they're social predators, so they will swarm their prey, and then they release enzymes that lyse the prey, breaking the cell open. All the cell's contents will spill out, and then the swarm will eat those contents. The swarm is actually able to behave as a single unit, even though it contains many different cells. Because those cells are all releasing chemical signals that tell the other cells like what's going on and help them act together as a single group. Once they have kind of found a prey, swarmed it, eaten it, then the swarm is going to continue like accumulating, and just kind of the bacteria will continue like moving in towards a center and aggregating, and they're going to end up forming a mound. And ultimately, from that mound, they'll produce fruiting bodies, which are interesting because they're actually multicellular. Then those fruiting bodies will release myxospores that are actually resistant to a lot of different environmental conditions. And later on, those myxospores, each one of them will turn into a new regular bacterial cell, which would be called a vegetative cell. So here you have an example of a cytoplasmic predator entering a prey cell. This is an example of a paraplasmic predator entering a prey cell. This is a colony of Myxobacteria, It has this strange appearance because of all the bacteria swarming together. They kind of end up forming these like ripples on the agar. And then here you have the fruiting bodies that are formed by the mound they make after they have eaten their prey. Next we have bioluminescence. Bioluminescence means light that is generated by an organism. There's certain animals that we think about as being bioluminescent, like certain fish, but actually those fish are not bioluminescent. Instead, they're living in symbiotic relationships with bacteria that are bioluminescent. You also have, like, not all bioluminescent organisms are bacteria, but they are all microbes, at least as far as I know. So you have some eukaryotes also that are microbial, that are bioluminescent. For the prokaryotes, you have bioluminescence in the bacteria. All bioluminescent bacteria are going to be in class Gamma proteobacteria in the phylum Proteobacteria, and you find them living in the ocean. Some of them will live kind of alone in the ocean, or, not like alone, but with the other bioluminescent cells. But some of them will be living in symbiotic relationships with fish or with cephalopods. Cephalopods include like squids and octopi. So all those types of creatures that we think about as emitting light down there actually are not doing it themselves. They have special organs called light organs, and inside of those organs you have bioluminescent bacteria living that are actually producing that light. So in these images here, these are colonies of bioluminescent bacteria. This is a fish with bioluminescent bacteria in it, in its light organs. This is that same fish photographed with the flash, and then this is that fish without the flash. So all the light in this picture is being produced by the bioluminescence itself. Here you can see some more bioluminescent fish, and this is a section of a light organ in one of these fish, and you can see the bioluminescent bacteria living inside of it. In order for a bacterium to be luminescent, it has to have the enzyme luciferase, and that enzyme requires oxygen in order to function. So these are all going to be some type of aerobe. The luciferase gene is actually super common to run across in the lab. It's used as a reporter gene. So basically anytime that you're kind of manipulating genes in your whatever organism you're working on, probably some type of bacteria, probably E. coli. Whenever you're manipulating genes in that organism, if you're kind of like adding new genes, in order to check that you added them successfully, you'll frequently also add luciferase, and then if you added them successfully, then that organism is going to become luminescent or bioluminescent. The expression of luciferase is controlled in an interesting fashion. It's controlled by something called quorum sensing. It's also called autoinduction. Basically, it means that the gene is only going to be expressed and used to make protein if you have enough bacteria present, only at a high population density. So they're all all the bacteria living in a column of bioluminescent bacteria are kind of releasing these chemicals that will signal to the other bacteria how many of us are here. And if there's enough of us here, then they all start expressing luciferase, and expressing that gene, making the enzyme luciferase, which allows them to create the light. The purpose of doing it that way is so that they don't make light when there's not enough of them for anybody to see it anyways. So they're not really making this light for each other. They're making it for animals who would be able to see the light and respond to it in some way. So you can't, as an animal, you cannot see the light that's produced by a single bioluminescent bacterium. There has to be a lot of them there for you to actually see the light. So that's why they're only going to make it when there's enough of them there for you to see it. So next we'll talk about morphological diversity, which is a part of functional diversity. So basically the different shapes and different structures that bacteria can have. Of course, we're not going to go through all of them, and we already mentioned some of the membrane invaginations that different microbes will have for photosynthesis, or for methanotrophy, or stuff like that. So we won't go through literally everything, just some of the things. First, we'll talk about the spirochetes, which are bacterial cells that are shaped like little corkscrews. So it's a real tight spiral. You also have other bacterial cells that are shaped like looser spirals. Those are called spirilla. So it's only the really tight spiral that's called the spirochete. All spirochetes are found in a single phylum spirochetes. They will live in different places, in the water, in the sediments, and some of them live inside of hosts, animal hosts. They all have what's called an endoflagellum, which is a flagellum that's located in their periplasm. So these are all going to be gram-negative bacteria that have the periplasm. So it'll be underneath their cell wall, but above their cell membrane. So it's kind of an internal flagellum, which is why it's called the endoflagella. The endo there means like internal. When the endoflagellum rotates, the entire cell will also rotate, which means it's going to just kind of spin around, almost like the propeller of a boat or something, and that actually allows it to tunnel. So it could be tunneling through the sediment, or more creepily, if it's trying to get into a host animal, it can literally tunnel through tissues. Thankfully, most of them do not do that to us, but there are some of them that actually do. There's 3 genera specifically that usually live in animal hosts, which would be Treponema, Borrelia, and Leptospira. Out of that, Treponema and Borrelia would be the ones that have species that we more need to worry about. Now, some of those are not like bad for their host. Some of them are mutualists, where they help the host. Some of them are commensals, where they don't do anything to the host either way, good or bad. And some of them are bad, which would be parasites. So for the parasites that affect us, you have Treponema pallidum, which causes syphilis, and different species of Borrelia, which cause disease. Those are usually transmitted by tick bites. A lot of these guys are kind of fragile. They can't take being in the environment, so they have to be in a host. So they would be transferred directly from person to person in the case of syphilis, or from tick to person in the case of Borrelia. So here's an example, like a drawing of a spirochete with the endoflagellum. So here you have your cell wall, and then the endoflagellum is underneath that. And you can kind of see it making like a ridge that runs along the length of the cell. And when it spins, this whole cell is going to spin. And then just some examples of the spirochete shape. So next we'll talk about budding and prosthecate microbes. Budding is a type of cell division. So most bacteria will divide by binary fission, where the cell just gets twice as big and it makes a wall in the middle, and then that wall kind of pinches off and separates the 2 new cells. With budding division, it's a very different process, where basically a new cell will kind of grow or bud off of the parent cell or the mother cell. So this is a diagram of that process. So in green you have your mother cell, and then in orange you have the daughter cell that's growing. So first you would start growing just a little extension off of the mother cell, and then you'll form a bud at the end of that. The replicated DNA, the newly replicated chromosome, is going to be transported down into that bud, and then the bud is going to just keep getting bigger until it's, the size of a new cell, and then it breaks off. So there's a number of different bacteria that use budding division. Usually bacteria that have those complicated internal membrane systems will be using budding division, because it just makes it a lot easier to build those systems. It's kind of hard to divide by binary fission without disrupting your complicated systems of internal membrane folds. Then you have prosthecae, which are kind of large extensions coming off of the cell. Not flagella or phyla, but things that are bigger than that. There's several different types. You can have stalks, you can have hyphae, and you can have appendages. In the pictures you see kind of different examples of them. So these are all counting as prosthecae. Even these are counting as prosthecae. And for these bacteria, here you can see a flagellum and compared to the Prostocae. So the Prostocae are much bigger than flagella are. Different Prostocae will have different functions in different species, but kind of a common function for them to have is to increase the surface area to volume ratio for those cells. So it's basically giving you a lot of extra surface area without giving you a lot of extra volume, so that you'll have more surface area compared to your volume. The point of that is that, the cell membrane is where you're taking nutrients across. So if you have more surface area, more cell membrane, you have more places where you can take nutrients up into the cell. Also, if you're making a lot of waste, that gives you more places to drop waste off out of the cell. So you're going to see Prostocae commonly on bacteria that live in kind of nutrient-poor environments where they really need to take up a lot of nutrients and have a lot of different surface area, available to take up whatever nutrients are around. Another function for the Prostocae is to attach the cells to different surfaces. That's pretty common. And then other functions as well that are more specific to the species in question. You also have some microbes that will make sheaths, which is like a casing that goes around the cell. It's like a cylindrical casing around the cell. It can get bigger than the cell. And some cells that will form into filaments will be sheathed, and this sheath is basically kind of forming the structure of that filament. So you can see that here, these are filaments. Filamentous bacteria, where it's just cell after cell after cell on end. Some filaments are not sheathed, but filaments like this are sheathed. So actually this whole thing is covered in this cylindrical sheath, which when you zoom in, you can see right here. So it's cell after cell, all stacked up inside of this sheath. The sheath is basically helping them hold together into that filament. And here you have kind of a medium view. This is the sheath, and then you have cells inside of the sheath. And where it gets kind of fuzzy here, this is a cell that's actually trying to leave the sheath, depart the filament, and go to a new place. So a lot of the time, bacteria that are living in filaments that are surrounded by those sheaths like that will have multiple life stages. The vegetative cell, which is kind of the normal cell, is going to be, living in a filament in a sheath, the sheath holding it together in that filament. And then whenever things get kind of bad there, like maybe nutrients are running out or something's going wrong in the environment and it's not a nice place to live anymore, then you're going to have cells at the end of those filaments growing flagella and becoming swarmer cells and leaving the sheath. So that's what's happening right here. This is a swarmer cell that is leaving the sheath. And if you zoomed in on that guy, you would see the flagella that he has grown. And finally, the last thing we'll talk about is magnetic microbes. So most magnetic microbes are bacteria in the phylum Proteobacteria, and all of them have magnetosomes. A magnetosome is a structure made out of membrane invaginations, foldings of the cell membrane, where they're storing those magnetic minerals that they're using to let them sense the magnetic field. The magnetosome is always oriented in a particular way with regard to the flagella of the cell, and the orientation is actually opposite for bacteria that are living in the northern hemisphere versus bacteria that are living in the southern hemisphere. So in this image here, you can see the magnetosome of a magnetic microbe. The magnetosome allows these microbes to orient themselves according to the magnetic field of the Earth, and then it lets them do magnetotaxis, where they're able to move in a specific direction according to the orientation of the magnetic field. Most of them are anaerobes or microaerophiles, so they're going to be living in the water or in sediments right where the oxygen zone or the zone of oxygen presence is ending. Especially for the microaerophiles, they really need to live just in that location where you have still a tiny bit of oxygen but not very much. If they go too far down, there's not going to be oxygen for them to do aerobic respiration. If they go too far up, there's going to be too much oxygen and they'll die of the toxicity from the ROS. For the anaerobes, they just basically need to go down below the oxic zone and not be above it, or they would die. So they're basically using this magnetotaxis to make sure that they are at the correct depth in the water or in the sediment, so to help them avoid oxygen. So they're going to use the magnetosome to orient down and then swim in that direction using their flagella. So instead of just swimming in a random direction and kind of hoping that they're going down, they're using their magnetosome to make sure that they're going down, and that's magnetotaxis. So we usually think about, if you're orienting according to the magnetic field of the Earth, you're either pointing like north or south, right? You're orienting according to the poles. But actually, the magnetic field also kind of points up and down. So as you can see in this diagram here, you have these magnetic lines coming out from the poles and going out into space before wrapping back to the next pole. So overall, the lines are going, from the north to the south pole, but they're also going up and out into space and then coming back down to get to that next pole. So if you're a microbe living in the soil or the water, it's easy to sense the part of this field that's pointing kind of up and down, and then orient down using that part of the magnetic field. And then you swim down and you go to a place with less oxygen, which is a place you like better.